This lesson is going to take a look at gene expression, so how we go from having our unique or particular genes to having appearance or behaviours expressed inside of an organism. We're going to look at cell differentiation, so how stem cells turn into particular cells with particular structural features and particular roles, and then also DNA methylation, which is just one of a few processes behind this switching on or switching off of certain genes. So the first thing I want to look at, just make sure I've got black. First thing we'll look at is the pathway from our genes turning into actual observable or noticeable features inside of our bodies. So we start off with our genes. So we'll put down our active genes. So these are the genes that we possess, that we inherited, and ultimately genes that have been switched on and are going to be expressed. So we'll look at how that comes about in a moment. So we've got our active genes. And the genes that we have, the genes that we are expressing, we refer to this as our genotype. So the sequence or the particular, what we call like combination of genes that we have inside of our bodies, we call that our genotype, our genetic makeup, our genotype. So these genes can lead to, will lead to the creation of proteins of two main kind of categories. We're going to have structural proteins, expressed and made through protein synthesis, or we might see proteins like enzymes and hormones. So more functional proteins, proteins that have jobs or roles inside of our bodies. The structural proteins, these are going to be what leads to our appearance. So the reason why we have certain hair colors, certain body shape and structure and things like that is because combinations of structural proteins have been expressed inside of our bodies, so it gives that certain look. The enzymes and the hormones are going to be responsible more for behavior. So because these have particular functions inside of our body, their functioning can give rise to the behaviors that we see. So whether we are kind of adrenaline seekers and we want to go bungee jumping and all that kind of stuff, or whether we prefer to read a book at home in the quiet, could be dependent a little bit upon our combination of peptide hormones and their activity and things like that. Now all of this, I guess we could say as well, as a little side note, can be influenced by environment as well, and a number of different environmental factors. So our genetic makeup is referred to as our genotype. The appearance and the behavior that we are actually able to see and observe is referred to as our phenotype. So that is our appearance, and that is the way that we act or the thing that we do. So the next kind of step beyond the expression of genes into structural proteins and enzymes, giving us that certain phenotype, is the influence it has on cells and cell differentiation. So part of that process of giving us a certain look or appearance is going to be because those structural proteins or particular enzymes that are switched on or off, expressed or not expressed, are going to give rise to the makeup of particular cells inside of our bodies. So most people are familiar with stem cells, the term stem cells, and have an understanding of the fact stem cells are our baseline cells when we are growing, when we are being developed as a child. We start off as this whole bunch of stem cells that pretty much can become any type of cell if we get certain genes expressed. So to simplify this massively, let's imagine that in this stem cell, genes A, B, and F are turned on or activated. This may lead to the development of a nerve cell. That exact same stem cell, if rather than A, B, and F being turned on, we saw genes C, D, E, and H being turned on or activated, this may have given rise to the creation of a red blood cell. So each pathway was possible because every stem cell has an identical copy of our DNA. Each nerve cell and red blood cell created from stem cells would have a unique copy of DNA, but it's just whether those particular genes are activated or left inactive. Once we get a nerve cell, or once we get a red blood cell, we cannot go back in that direction. So we are not able to go back to a stem cell. Once the particular type of cell and the cell's function and the structural proteins and the enzymes within and all that kind of stuff has been set, then that is what it's going to stay as. Now, one of the features, or one of the processes, which controls whether we see genes being switched on or off, so genes being expressed or genes not being expressed, is something called DNA methylation. And it involves exclusively cytosine. So we know that cytosine is one of our 4 bases found on DNA and can be attached to sugar and phosphate in order to make up a nucleotide. So DNA methylation involves cytosine and involves something called methyl groups. So this here is a methyl group. Now, cytosine, our regular cytosine, is pictured here on the left, whereas this picture on the right is something called methylated cytosine. So the adding or removing of these methyl groups from cytosine can have an impact on whether we see genes being activated and then expressed, or genes being left inactive, or we actually call that, the kind of proper term is silenced. So genes can be silenced, which means they aren't expressed, or genes can then be activated, and we'll see genes being expressed, which will give rise to a certain structural protein, which will give rise to a certain appearance or phenotype. So if I was to draw a sequence of DNA, and I won't draw nucleotides for the purpose of time, we'll just draw a sequence simply like so. So I've got a little sequence there, and what we will see is either that sequence will be left as is, or if there are methyl groups on the cytosines and we want this gene to be expressed, what will happen is the methyl groups will be removed, and we will see that the gene without methyl groups is going to be expressed, and we will get the structural or functional proteins being created from that gene. Transcription and translation is able to occur. Now, if we don't want this gene to be expressed, something called an enzyme called DNA. Let me just make sure I spell this right. Methyl transferases, methyl transferases. I think that's right. So these enzymes called DNA methyltransferases, if we don't want this gene to be expressed, they're going to come along and they're going to add these methyl groups to the cytosines. So these DNA methyltransferases, just get rid of that, get that out of the way, they will come along and they will add methyl groups, just CH3. They will add methyl groups to all of the cytosines. What this will do is effectively switch this gene off, and this gene will be silenced and not expressed. So the silencing or the expression of certain genes is what's going to give rise to certain structural and functional proteins, and in turn that's going to lead to different types of cells being created, or different appearances being given, different behaviors being given in a particular organism, and we call that the phenotype. So the genotype, the sequence of genes and which genes are being activated, leads to a particular phenotype in an organism.